Okay, I think we'll get started. It's right at 12:30. So thank you for joining the North Central IPM Center Pests in Progress webinar series. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. The North Central IPM Center is one of 4 regional centers supported by USDA NIFA Crop Protection and Pest Management program. We're on a mission to coordinate IPM across the region by sharing and promoting IPM research, extension, and teaching activities. During the webinar, if you have any questions at any time, please type them in the Q&A part at the bottom of your Zoom screen, and our speaker will answer the questions at the end of the presentation. So again, please use that Q&A. It's a little bit easier for us to follow there than if you put it in the chat. Today's webinar is the ecology epidemiology of the invasive longhorn tick ecological epidemiology of the invasive longhorn tick insights from Ohio. And we're excited to welcome Andreas Eleftheriou from the Ohio State University. So I'll let you take it away, Andreas.
Great. Thank you so much for that introduction. And thank you everybody for joining in today. You can hear me just fine, right? Laura, everything good? Okay, great. So so I am a veterinary public health resident at the Ohio State University. So I'm stationed at the College of Veterinary Medicine. And today I wanted to talk to you a little bit about this new longhorn tick that we're concerned about spreading across the eastern United States. Now, the name of this tick is currently Asian longhorn tick, but there are discussions within the community to change the name to longhorn tick, so taking the Asian out of the name. So just a heads up, in a few months, if you do end up seeing longhorn tick without the Asian, it's still the same tick. So keep that in mind. Now, I will focus a lot on the findings that were generated from a study that was funded by the North Central IPM Center as a critical issue study. So thank you very much for that funding. But before I do that, I will introduce you a little bit to the Asian longhorn tick. So let me go to my next slide here. Once we talk a little bit about the Asian longhorn tick, just to get you acquainted with this particular tick, we are going to move on into the situation currently in the United States in terms of where this tick is and where it's going. And I'm going to start my story about what's been going on in Ohio and how we got started with this surveillance project that I'll be introducing you today after the outbreak that occurred. And then I'm going to finish off with some conclusions and really some take-away messages that will be useful for integrated management. So the sort of approach that we took here with this study is to really understand ecology and epidemiology first as sort of foundations for driving an integrated management for this particular tick in the future. All right, so let's first start with understanding where this tick is from and where it's gone. So this Asian longhorn tick has a native range in East Asia. So if you're in this map, you can see that all these red dots up here in China and South Korea, like those are essentially representative of reports where this tick has been found. So essentially that is its native range. And eventually it ended up finding its way down to Australia and other islands within that region. So we got New Zealand and other Pacific islands. So it's thought that it was introduced to that region via the livestock trade. So there were probably ticks on cattle and they were not noticed. And then it found its way to the United States. As you can see here with all these red dots here in the eastern part of the United States. And now the United States is part of the introduced range of this tick. We don't know exactly how it got here, but we know that it officially was identified as being outside of a U.S. port of entry in 2017, although there's some discussions that maybe it was detected back in 2010 and misidentified as another tick. Regardless, we started this surveillance of this particular tick, like the whole community that works on this tick in 2017 and has been spreading since then. Now, this is what this tick looks like, just to give you a visual. You may be familiar with some of our native ticks, such as the black-legged or deer tick, which carries Lyme disease. So it looks a lot different from that tick. It's actually a bit larger. So here what you can see is going from the left to the right is essentially the different life stages of this tick. So typically these types of ticks, hard ticks we call them, such as the black-legged or deer tick, they have three life stages that look for a host to get a blood meal. So here I'm showing you that we're going from larva on the left, which is sort of the baby tick, moving on to the nymph, which is the teenage tick, if you will, and then we end up with the adult female. And you can see that they vary in size and in how they look after they ingest blood. So I think the visual is pretty strong when we look at an unfit female on the right side and an engorged female. Their size differs, so it can become also difficult to identify them because they blow up. So it's really hard to ID them under the microscope when they're engorged like this. Now, this is just down here in the bottom left. I'm showing you just a picture to demonstrate how small they are and the nymph size when you compare it to a human fingernail. So you may be wondering, well, where's the adult male? You're not showing me any picture of an adult male. Well, it turns out that in introduced populations, so Australia, New Zealand, and now the United States, males are very rare in the population. And this is because females can reproduce without mating with any males. So they can go through asexual reproduction. And females can lay up to 2,000 eggs. Now, none of our native ticks can do this, as far as we know. So essentially, these female Asian Longhorned ticks can clone themselves. So you just need to get one female in a good habitat and she clone herself. And then those females will lay 2,000 eggs and so forth. So that's why this tick can reach such massive infestations very quickly, because it does not require sexual reproduction. All right, just a few key things here about the biology of this tick that I want you to be familiar with. This is a three-host tick. So what that means is that those life stages that I mentioned in the previous slide, each of them needs a blood meal from a different host. And this is very typical of many of the ticks that we are familiar with, whether it's the American dog tick or whether it's the black-legged tick. All of these ticks require three different hosts. This is also a tick that is a host generalist. And what this means is that it doesn't really have a specific species or animal that it wants to bite to get blood from. It is open to feeding on humans, domestic animals, and wildlife, and actually reports of tick bites of this particular tick coming from humans and all sorts of animals, wildlife and domestics, have already been recorded in the United States. So this tick has been busy. Particularly, it seems, though, that even though it's a generalist, it seems to prefer medium and large-sized mammals. So when we think about wildlife, that will include our deer, white-tailed deer seem to be a preferred host. And when we think about our agricultural species, cattle seem to be very preferred. Now, what I'm showing you here is a schematic of a life cycle of this tick taken from the CDC, because we'll talk about how this tick also has human health implications. But what I want you to take from this complicated diagram is that this tick can take up to 3 years to complete its life cycle. So it's not a tick that might finish up its life cycle within a few months. There's seasonality and in its different life stages and when they show up in different habitats. So it's just good to know that it seems to have that life cycle timeline that's very similar to a lot of our other native ticks that we're familiar with. All right, so why do we care about this tick now that it's spreading in the eastern portion of the US and potentially elsewhere in the US? Well, it's a significant pest and disease vector. So this is a major pest of livestock, and especially in introduced ranges. And it's primarily a livestock pest of cattle and sheep. Those seem to be very important agricultural species that are affected. This species, the tick species, can show up in such massive infestations as you see in this picture here from an agricultural animal, to the point where you have all these ticks consuming blood from this animal, and they can reduce the blood within this animal, and eventually this can cause death. So we know of mortalities that occur in agricultural animals just because of heavy infestations. It's also important to realize that these animals can, even though they might not die, they might have reduced growth and reproduction. So this tick is associated with economic losses in agricultural species. Particularly, we know of studies that have detected this outside of the US, because obviously this is a newcomer in the US, so we still don't have very good numbers about economic losses, but it's anticipated that it might be very similar if we don't control it appropriately. Now, this particular tick species, as I've mentioned, has implications for human health, but also agricultural health. In particular, we know from just in the US, we know from laboratory studies that have been done that this tick, the Asian local tick, seems to be good at transmitting the agent for Rocky Mountain spotted fever and also a number of tick-borne viruses, such as Bourbon virus and Poisson virus. Now, no reports outside of the lab have come that, let's say, some animals or humans have been infected with these pathogens because of the Asian Longhorned tick. However, it's something that we're monitoring because we want to make sure that if this does occur, that we need to be focused that, okay, these pathogens are coming from the Asian Longhorned tick, so control needs to be addressed on this particular tick. But the big one, the big pathogen that we're concerned about for the cattle industry and for agriculture in the US is the parasite Thaleria orientalis, which causes bovine theileriosis. Now, this is a parasite that shows up in red blood cells. So this is something that might be hard for you to see, but these are red blood cells, a blood smear from an agricultural animal's blood. And where the circles are, the black circles, that indicates an infected red blood cell. And this makes his red blood cells want to break apart. And then you end up getting an animal that doesn't have enough red blood cells to oxygenate themselves. So you can imagine that this can lead to anemia, unthriftiness, reduced body weight, and eventually can cause death if these animals lose too much blood. So this is the big one that we worry about in the cattle industry. And we know that there have been outbreaks of bovine theileriosis in Virginia and a lot of nearby states. And we have had a detection of this in Ohio as well. So it is one of those pathogens that is actively spreading with the Asian Longhorned tick, and we believe the Asian Longhorned tick brought it with it. So it's sort of a foreign animal disease that's now spreading through our cattle industry. So it's something that we are really focused on in terms of making sure that we control the Asian Longhorned tick so this particular parasite doesn't keep on moving and spreading to naive herds. All right, so now that I gave you a little bit of background, I want to launch the poll if possible, because I'm curious to know about your answer to this question. So Ohio is the only state within the North Central IPM region that has reported infestations with the Longhorned tick. Now, do you think this is a yes, no, or are you unsure about this particular statement? So we'll wait a little bit until you are able to answer this question or this tell us what you think about the statement. And let me know how I can access the results when most of them seem to have answered the question. Okay, good. Okay, so it seems that we have educated folks in our audience. So you are correct that Ohio is not the only state. So I want to show you here a map from the USDA APHIS situation report. This is the most updated map, and USDA APHIS actually has a national monthly call where folks from different affected states can participate, and we learn a lot about surveillance that's going on in different states to understand where it's spreading. But this is the map here. Don't worry so much about the colors, but what you see each year in a different color is the county of that state. So we can see that a lot of the affected counties are within the Mid-Atlantic region. So we see that originally they started in New Jersey and it's spreading upwards and it started spreading into Virginia and it's going further south. And now in Ohio, we are at 11 counties that are impacted by this tick. You can see that Indiana has had a particular county that's been affected, and then Missouri as well. Now, as you can see how this tick is spreading, it is concerning about whether it's going to keep on moving towards the North Central region. So that's why it's important to understand how this tick interacts with the different environmental and ecological factors that we have in Ohio and in North Central region at large to understand how we can control it in our region, because it seems to be coming towards our direction. All right, so now I'm going to dig into the ALT invasion situation in Ohio, because Ohio is really that state in North Central region so far that's been mostly affected by this tick and can potentially serve as a model for the rest of the region. But what I'm showing you here is a figure from our paper that was published in 2023 that was describing the original outbreak that occurred in a cattle population in Ohio, which is when we were alerted at the Ohio State University that this tick was spreading and establishing populations in Ohio. So this is just West Virginia and Ohio. We believe that the tick is coming from West Virginia. It makes logical sense that that is the case based on this map, on the map. But what I'm going to be focusing today is on my study location, which was in this county right here that borders West Virginia and Ohio River is actually separating West Virginia from Ohio at this location. But the other thing I want to mention here is that this map, if you ignore the stars, this map essentially was describing what was going on in 2020, of July 2020, sorry, in July 2021, July 2021. So the stars show the most updated situation in 2024. So we can see that it's spreading further into Ohio, unfortunately. All right, so now I want to walk you through the timeline of the investigation of this outbreak, which essentially ended up being the site of the first known established Asian local tick population in Ohio, which we described in our publication. And there are links that I provided to the facilitators that will take you to that publication if you want to read more about it. But here, what I want to do is walk you through this because this is the timeline and the initial outbreak that motivated me to apply for funding through North Central IPM Center to study what was going to occur after the outbreak. So in July 2021, there were 18 cattle infested with Asian Longhorned ticks, and 3 of them were found dead, including a bull, and 15 were isolated and treated with porrin permethrin. So it was very surprising that 3 of them were found dead, presumably because of just blood loss. They had lost so much blood that they had actually perished from heavy infestations. So the producer ended up mowing the pasture that these animals were on. And then they returned the cattle, but they became reinfested with the Asian Longhorned tick. So even though it was mowed, the pasture, the ticks were still there. So the cattle were again isolated and treated with porrin permethrin. And the Ohio State University and USDA APHIS Veterinary Services were contacted to come in early August 2021 to the site to collect samples to identify the degree of infestation and how many ticks were going on, like how many life stages and what kind of tick we were dealing with. So over 90 minutes, our group collected 479 adult females, 5 nymphs, and 8,803 larvae. So this was indicative of a massive infestation that was occurring at this time point, because it was even over 90 minutes of collection. So, okay, we were realizing, okay, this is the first really huge outbreak that we're getting here of this Asian Longhorned tick, first time in Ohio. Now, the Asian Longhorned ticks are so many that started spreading to neighboring turfs of human residences that were living near the pasture. And because it was encroaching into human environment, they were able to get a licensed pesticide applicator to apply lambda cyhalothrin, which is a pyrethroid, to the turf. And that seemed to be successful enough that got the producer motivated to apply this particular pesticide to the pasture. And that is when the producer felt confident enough to return the cattle back to the pasture in late September 2021. And then by the end of November 2021, when the grazing season ended, the cattle were removed. There were no problems with ticks after that multiple pesticide application on pasture and treating the animals as well. Now, we weren't sure when we were consulted about this, we weren't sure if it was the integrated management, like the mowing of the pasture and the pesticide application on pasture and the animals that was really making these ticks like not be active anymore, or whether we're getting close to cold weather. So they were likely to become inactive by the time November comes with cold weather. So this is why we were sort of at this stage and we're like, let's hopefully get some funding to continue on studies to figure out what's going on with this tick after this outbreak had ended. Before I go into that story, I want to mention here that we tested Asian Longhorned ticks for pathogens that have veterinary and medical significance. So we tested 100 adult females from that initial outbreak. And 8% of them were positive for Anaplasma phagocytophilum. So this is a disease agent that causes anaplasmosis. We didn't go into detail to find out exactly if it's the one that causes human disease, because there's different variants. But the fact that it was able to transmit this, because these were ticks that were looking for a host. So if they were infected and were looking for a host and could bite on a host, they could transmit this parasite or pathogen to a host. Thankfully, all the ticks were negative for all these different other disease agents, including Theileria orientalis that causes bovine tuberculosis. So we felt confident then when we made the assumption that those 3 cattle, including a bull, had died, it was likely because of massive infestation, not because of an infectious disease exposure, such as bovine tuberculosis, because we weren't finding this particular disease agent on site. And the cattle, as an FYI, there was blood collected from the live cattle, and they were negative for Theileria orientalis, just to make sure. All right, so here now we're getting into the objectives for the critical issue study that was funded in 2022. So we had 2 objectives. We wanted to characterize the ecology of this invasive ALT through active surveillance of the environment and wildlife. And we wanted to describe the epidemiology of associated disease agents, just like I showed you in the previous slide. All right, so let me just quickly go through some of the methods that we use. What I'm showing you here is a satellite view of the study site. So you can see this whole area here, this was the pasture where the animals were in. But what happened is that at the end of 2021, and we came in 2022 to start our study, the landowner had decided to not allow cattle back on. They decided to just have the pasture transition into a cropland. So this actually had no cattle anymore, no the pasture. So we didn't have cattle around to sample or anything like that. So it moved into cropland. But what I'm showing you here is that we had different habitats available at this site to look at. We had grassland, forest, particularly a lot of forest surrounding the now cropland, and edge habitat, so that ecotone zone between forest and other types of habitat. So we use 1 meter square muslin cloth. You see me here dragging this muslin cloth behind me because ticks that are looking for a host will grab onto this muslin cloth as it passes by. And then we sample 3 habitat types because we wanted to understand habitat associations in Ohio. And then we sampled once a month starting from May until November. And then we also sampled wildlife because we wanted to understand host associations. So for the wildlife live trapping, we had 1 session per month from June to October. And primarily we focused on small mammals in forests, collected ticks and released them. And then we also did some opportunistic sampling of wildlife where we collected any roadkill that was nearby, that was fresh, and we could look for ticks on them. And also we examined a harvested white-tailed deer at nearby stations in early December of 2022. So we collected ticks from these animals. Now we're ready to go into our results. So this is showing you that the phenology of this tick, so essentially how it changes over time in its density, resembled really what we saw from mid-Atlantic United States studies, because we had a lot of literature in that region, but we didn't have anything in Ohio or really anywhere else in the North Central region. But there were some minor differences. So what I'm showing you here is that when we were out in May, we didn't catch anything, like no ticks. And then June comes, and you can see that we have a peak in nymphs. So it seems like nymphs are emerging and peaking in June. So then in July, we started seeing a lot more adult females. And then as time went on, in September, we got a huge peak in larvae. So this phenology that you see here is very similar to what we see in mid-Atlantic. But there were the couple of differences, however, was that in September and October, in other studies, there's more of an overlap with adults and nymphs. Here, we were just seeing a bunch of larvae. And then also in other studies in the mid-Atlantic US, and when I say mid-Atlantic, think about New Jersey, sort of Virginia, Maryland, like those particular regions. The phenology started in March. So they started a whole lot earlier. Now, I know what you might be thinking. Well, there was integrated management done the year before. And you're right, there was integrated management done the year before. And in these other studies, no management had been done. It was all naturally occurring densities. So we couldn't tell if this was like just the norm for Ohio, or it's really the after effect of integrated management that occurred the year before, which is likely if you think about it. So it could be that they're showing up in June because there was integrated management done the year before, which is obviously a good thing if we're actually delaying when they're showing up because we're doing some sort of management there that's successful. But this is the phenology data that we had accumulated from the study. We also found that there were some differences in terms of the habitats that they were associated with. So, for example, when we look at the grassland here habitat, it seems that we were just seeing a lot more larvae in grassland, and we were seeing a lot more. Really, we just saw adult females at the edge, at the ecotone region between the forest and cropland. So that's where we were seeing a lot of our adults, which potentially has implications for management if we were to be targeting only our adults. But then our nymphs were found in forest and grassland, but didn't seem to be found in the edge habitat. So there were some habitat associations there. A really important take home here, though, is that we found them in all habitat types. So unlike the black-legged tick, for example, that when we were doing the study, we found tons of black-legged ticks in the forest, but we rarely found them anywhere else. So this tick is in contrast to the black-legged tick, so the integrated management needs to target more habitat types than what you would think for the black-legged tick. And then lastly, we didn't find any Asian local ticks on any of the wildlife hosts we sampled. So in our live trapping data, we focused a lot on small mammals, nothing on small mammals, like nothing on mice, plenty of black-legged ticks, but nothing to do with Asian local ticks, which actually corresponds with studies in the mid-Atlantic US that rarely find anything on small mammals. And indeed, they find more Asian local ticks on larger animals, such as raccoons, skunks, possums, and those are typically the range of where they found them in, the sort of the mesomammals, if you will. In our opportunistic data, again, we didn't find any Asian local ticks, and we sampled 51 white-tailed deer, and that was in early December of 2022. We were surprised by this because we were finding black-legged ticks, and we were finding the moose ticks or the winter ticks, but no Asian local ticks. However, this was after. Remember how in November we stopped finding any Asian local ticks in the environment? So it's likely that we missed that overlap. So by the time December came, the Asian local ticks were no longer questing. We also tested again 41 adult females and nymphs for different agents, and we found this time again Anaplasma phagocytophilum still relatively low prevalence, but still there. It's still persisting. Even though integrated pest management was done, Anaplasma phagocytophilum still persists in our tick population, but thankfully still negative for everybody else, including Thaleria orientalis. So that was good news that at least we're not picking up anything else. But do notice that we ended up having all the seasons, right? We ended up having only 41 that we were able to collect from the environment compared to 100 or more before with the initial outbreak. So that pest management that was done probably was helpful in reducing what kind of ticks we were seeing, the tick densities. So now some key takeaways that are relevant for IPM regarding this tick. We found that ALT life stages were active June to October. Might start a little bit sooner if we didn't have integrated pest management done the year before. We found them occurring in grassland, edge, and forest. That seems to be in contrast with a lot of our native ticks. And we did not find them on small mammals, which tend to be disease reservoirs for a lot of different types of human and veterinary diseases. And indeed, we've really come to the consensus that, yes, this is good information that we now have as sort of a preliminary sort of study to guide other studies. But I think we're going to need more similar phenology studies in the North Central IPM region to refine our findings and to guide management approaches within our region. This is a list of useful resources that I have here on the APHIS USDA website that are very good at seeing what's going on with this tick in the United States. And I am happy to take any questions. If you have any, thank you all for joining in. This is my email address if you have any follow-up questions that we might not get to during the webinar.
Thank you so much. That was very informative. So I know you just shared a bunch of resources and this is a really dynamic system. While we're waiting for people to type questions in the Q&A, like where's your kind of best place to stay up to date regarding like, new places where Asian longhorn tick is being found or transmitting diseases? Like where's your go-to spot? Oh, you're muted. You're still muted, Andreas.
Thank you. You can see my screen though, correct?
Yes.
Right. Okay, great. So yeah, so this is a good list of sites on the APHIS USDA website to find out exactly what's going on. They have like nice story maps in terms of like updating the different counties that are being reported as positive from different states. So I found that very useful. There's also that national USDA APHIS call that I was mentioning that shows up every month. Like every month, we all meet different stakeholders from different states to discuss what's going on, and that's a good way of learning about what's happening in other states. Now, I believe anybody can join that, and I can, if anybody's interested in that, they can email me and I can get them in touch with Denise, who runs that particular stakeholder call. And that's how I stay up to date with a lot about the different things that are going on with this tick.
Oh, very good. That sounds like a good resource. One of the questions we have is why that particular pasture field in Ohio? How would the Asian longhorn tick have been introduced there?
Yeah, so we are sort of like dependent on our livestock producers. When they notice something wrong, something really wrong that's unusual for them, they will contact a lot of the time local extension agents, and they potentially could contact us as well at the universities level. And this livestock producer was finding something wrong, and they were like, "Oh, I need to find out what's happening. This is very strange. All these different little spider-looking organisms all over the place." So that's how we were alerted for that specific pasture. Now, it's what we are hypothesizing is that the ticks were somehow because they, as you were probably able to see from that map of Ohio and West Virginia, there were counties along the Ohio River on the West Virginia end that were positive for this tick. So what we're hypothesizing is that it came either through maybe a human or most likely a wild animal like a deer was able to bring it across into Ohio, and that's how we think that county, being at the border, was able to be affected by that tick expansion. So that's kind of our hypothesis. We never found out exactly what the answer is, but that's kind of what everybody else is considering.
Okay. What were the hosts for the ticks in Missouri or even the habitat? Like, about the Missouri sites, hosts and habitat?
The Missouri site, I want to say that it's I want to say that it was agricultural fields, sort of like pasture as well, pasture environment. I don't know entirely. There is a publication that I am aware of that talks about that report. So if people are interested, I can find it and share it with you. But there is a publication that came out, but on these different calls, they don't tell us exactly like what the habitat type is in these different states, but that information is out there in that publication, so it's somewhere available where it says exactly where they found the tick because it was the first time they detected that tick in Missouri, so they wrote a publication regarding the habitat and all that.
Okay, thank you. Another question, were you able to estimate the percent of Asian longhorn ticks compared to the other tick species you collected?
Yes, good question. So for the most part, we were collecting black-legged ticks. That was really we were collecting maybe some American dog ticks, very few, but for the most part, black-legged ticks in the woods with Asian longhorn ticks. And everywhere else, it was just Asian longhorn ticks outside of the woods. So it seemed to me that when we're looking at different proportions, it seems that Asian longhorn ticks are a bit more dense, higher densities than black-legged ticks. Now, whether there's anything going on there in terms of competition or any sort of impact of the pesticide pesticide that was applied to the pasture, did it affect the black-legged tick as well somehow? Everything's sort of like uncertain, but it does seem that Asian longhorn ticks seem very dominant at that site. And we don't know what was going on before. So it would have been interesting to have before data to see what was going on with the black-legged tick and any other ticks, but we don't have those now.
Another good question. So what is the recommended IPM practices that are effective against this tick? How can the spread be checked? I mean, do you know kind of what animals are on and when and the movement?
Yeah, that's a good question. And actually, USDA APHIS most recently put up a guide to how to to address tick control of this tick, and also different types of other guidelines in and a PDF online. And we were given that when we were doing the webinar recently. And if it's something a PDF, I can maybe send it to you, Laura. You can share with everybody else because I don't know if it's online, but we can distribute it to everybody. Like it's publicly available technically. So what's been said is that for this tech, you really don't have to worry about doing anything. Well, so far, the recommendation is that based on what we know, we can follow through and do what we are doing for our native ticks, it seems like. So it seems that you have to focus on treating the animals and also doing pasture management, whether that's going to be mowing lawn to keep everything very short, the grass, so ticks don't like the habitat anymore because they dry out when there's too much sunlight that goes through it and they get exposed to. And also another recommendation is to remove a lot of brush and a lot of areas where a lot of wild animals can congregate, especially deer coming through or skunks or raccoons on pastures. So making sure all that any log piles or anything like that are removed from the pasture to make it less likely for wildlife to come into the pasture. And essentially, the other portion of this is to apply the same pesticide treatment that's applied on pasture that for our native ticks, very similar for this particular tick. Now, any labels, pesticide labels that are currently for our native ticks, you're not going to find a label for the Asian Longhorned tick yet because there needs time to go through that approval process, but they're still supposedly effective against the Asian Longhorned tick, whatever we have for our native ticks in terms of pesticides.
Okay. Are we seeing any resistance in Asian longhorn tick or any other tick?
Yeah, so I can speak for Asian Longhorned ticks. This is something that came up recently. So as far as we know in the United States, there is no resistance being developed. This is an evolving situation. So as more spreading and pesticide application occurs, or that's why we promote integrated pest management. So we don't just use pesticides, we use other types of culture and physical practices. But as far as we know currently, no resistance has developed.
I guess there's no other question, but whether, just I guess overall, with the longhorn tick, I mean, are you expecting this to increase, be very problematic, fester at the levels it's at? Like, how concerned should we all be?
Yeah, so this tick is going to be spreading. I mean, it's here to stay, unfortunately. Like it's been originally there was the idea that maybe we can eradicate it because it's just focused in New Jersey and it's not moving to because this particular tick, obviously ticks don't move like too fast, so it needs wildlife to move to different places and livestock trade to move them even further away. So we were thinking at first that we could eradicate it when it was in a very localized place. But now because it's spreading all over the place and it has wildlife hosts. When it has wildlife hosts, forget it. It's so hard to control wildlife, and then it becomes very difficult to control ticks after they have wildlife hosts. So we shouldn't. I don't think we should be concerned per se. I think that we should be very aware, especially because this tick can clone itself. I think that's the important feature of this tick because no other tick can do this. So it's important to, if we do notice Asian Longhorned ticks, we need to be more prepared to apply treatment quicker than maybe we do with other ticks because of that cloning capacity. So we want to make sure we get to them before they have the chance to clone themselves even more. So I think that's my take home here, that to have a little bit more concern for Asian Longhorned ticks because of that cloning capacity. And we don't, and because of also the importance of it carrying bovine tilviosis and transmitting that disease. But otherwise, I wouldn't be super scared of it or anything like that. Like, I'm being attacked by another tick. It's going to be too many ticks all over the place now. Wouldn't be that concerned about it, but we do need to be a little bit more aware because of its cloning capacity.
will this be primarily cattle or what, any other humans, any other livestock?
Yeah. So reports have been coming in of this tick biting humans, biting pets, so dogs, cats, also biting cattle, biting sheep, all our domestic species, swine, chickens. So it will bite onto birds. Actually, it's been found on migratory birds as well in the US. So we're learning as more people do more studies and try to do active surveillance, we are realizing that it's found in a lot of different hosts and then becomes even more difficult to control. But it does seem to really like deer and cattle. It seems to like just accumulate on them and just get huge infestations on them. But otherwise, I would say the other species that have been noted as being particularly preferred would be raccoons, possums, and skunks. So are those mesomammals that happen to like being close to people, like in urban areas. So they could potentially bring it in into new areas.
Very interesting.
Yeah.
Well, I'm not seeing any more questions coming in. So I want to thank you so much for the excellent presentation. Thank the audience so much for great presentations, for great questions and attending. So I'll let everyone go. Just a reminder that our next webinar will be May 15th at 2:30 p.m. And we're going to hear from the Great Lakes Urban Agriculture IPM Working Group, and that'll be hosted by Maggie Rivera, also from Ohio State University. So we will see everyone in May. And thank you. Thank you again, everyone, and have a great afternoon.